So not to get in the way of, the moderator should lead off with a really intelligent question, but I don't want to get in the way of people that really know what they're asking. So please go right ahead. And would you like to shout, or let me take the microphone over to you? Which would you prefer?
It's okay, I'll speak loudly.
Okay.
Dr. Gregg, thank you for the very interesting talk. I'm curious, how would you inject the IG of Chimera into a patient?
So I've done those primarily IP in the mice. We've done them Okay. Is it working now? Yeah, there it is. Yeah, so I've done them in the animals, both IV and IP, and it's worked effectively both ways, so I would imagine probably IV would be sufficient as well. The nice thing about these molecules is that, unlike a lot of the other agents, they're not going to initiate any type of immune response. They're not going to inflict any type of fever or any toxicity. They have a nice 2-week half-life, so I think they're pretty safe to use.
Have there been any human trials to date?
Not as yet. I mean, like I said, I'm still in the preclinical phase, and these are the first rounds looking at cancer. I worked with the same molecule in terms of autoimmune disease. We modified it slightly to shut down responses instead of eliciting them, and that work actually has progressed to a pharmaceutical, and so there are clinical trials ongoing with those. Yeah.
For the advantage of our online attendees, if the panelists could repeat the question, or I'll be glad to repeat the question as well, just for clarity, but that's fine. Who else? Yes, please.
Yeah, also for Dr. Gregg, that was enjoyed your presentation. There was a report recently of a patient with cholangiocarcinoma, I think it was just in Science last week actually. A patient with
Chimera is what you're saying, and then infuse those back into the patient. That was actually one of the things I thought about doing first, and I thought, well, why should I go through the process of doing that ex vivo and then putting them in the animal when I just inject one injection of the animal and see? And so this was actually the first trial to see, could I get a good immune response? And I got a great immune response. So it kind of led me down a different path. But yeah, that was the first thought I had, was actually doing that. So it's a good point.
Great. Dr. Pledger.
That's a great question. The question is whether or not there was a selection process of the xenolines that are grown in animals, that since it takes 2 to 3 months for the tumor to actually grow up to a point where you can harvest it, as to whether or not there was a subpopulation of more aggressive tumor cells that were selected in the process. And the answer is that probably is occurring, but it doesn't occur as rapidly. The changes don't occur as rapidly as they do in tissue culture, where the entire microenvironment has changed. And so there you're selecting for cells that respond to high oxygen content. In other words, out here we're breathing 20.8% oxygen. In normal tissue, it's about 10% oxygen tension. In tumors, it's less than 1%. So tumor cells really have to struggle to get oxygen. The same for glucose. You put tumor cells in culture, and you put in 4.5 grams of glucose per liter of medium. If you had that coursing through your veins, you would be comatose. So essentially, it's around 1 gram or less of glucose per liter of plasma. And then the other issue is that, as you well know, and everything I know about growth factors I learned from you, especially about platelet-derived growth factor, is that the growth factors tend to drive a change in the phenotype. And those growth factors are not expressed to any great extent in the mouse. Platelet-derived growth factor, of course, is a wound fluid component. Fibroblast growth factor, transforming growth factor beta, all of those can promote a different phenotype by changing the gene expression profile of a cell. And in the mouse, that doesn't really occur. Now, what does occur is that you are selecting for a cell that can survive in the mouse. And the issue is, how closely does that resemble the cell or the cells that you took out of the patient. And so when we compare the patient's tumor with the xenoline after 5 passages, they're about 92% the same. The differences are basically things that people don't realize are in tumors that comprise tumors, especially tumors in the brain. There are a lot of normal stem cells, normal neural stem cells in the brain that migrate to tumors. They see tumor cells as a wound, and they go in to kind of repair the wound. There are also a lot of lymphocytes, macrophages, that actually migrate into the tumor for the very same reason. So those genes actually disappear from the xenolines because those cells are not in the mouse. Those normal neural stem cells or normal neural cells or human lymphocytes or human macrophages are not in the mouse. So they do change. So the genetic profile does change. But, you know, it's not bad that we're selecting for the most aggressive tumor, because what happens when you treat patients with drugs like temozolomide or with radiation is that you select a subpopulation of the meanest, baddest cells in the tumor. And essentially those are the tumors that kill the patient. It's not the original tumor that kills the patient. It's the tumor that comes back with a vengeance after you've hit it with a stick. And so essentially, the issue is that these tumors tend to resemble more of the patient tumors that recur than the patient tumors that were originally there.
I really like your idea about having the avatars, because we start and all that is
Well, a lot of the pharmaceutical companies now are moving towards avatars. The National Cancer Institute is establishing an avatar program where you can get them to test tumors for you. And a lot of the pharmaceutical companies are actually going to what is known as an N of 1. So we normally do mouse experiments with 8 to 10 mice per group, per treatment group. And the drug companies are using these avatars as basic screening. They're saying, you know, the tumor is the same from mouse to mouse to mouse. The mouse is the same from mouse to mouse to mouse. So why not just use an N of 1? In other words, just treat one mouse with whatever drug you're going to be testing, and that way you can look at 1,000 drugs and 1,000 mice and get some answers as to what actually works.
More questions? Yeah. Okay. Let's go this side, back this way and that way. Okay?
Yeah, also for Dr. Gillespie, you have that very nice response with the JAK2 inhibitor, and I think some other responses with some of the small molecule tyrosine kinase.
Right. So the question is, if you're using a mouse model, how well does it recapitulate the fact that the blood-brain barrier is one of the major problems for treating animals due to accessibility of drugs? So first of all, the JAK2 inhibitor we used has really good blood-brain barrier permeability, and that's the reason the company pulled it off the market, is that they were using it. They had gotten up to phase 2 studies in myeloproliferative disorder, but it was causing a lot of neurological pathology. And so they had to actually pull the drug because of the toxicities. But to answer your question in the broader sense, the tumors we use, as you saw in the slide, are more invasive. In other words, the tumor cells actually crawl out into the normal brain, where the blood-brain barrier is essentially still intact. It's leaky where the big mass of cells are located, but it's relatively intact, and that's the problem. All of these tumors come back within 2 centimeters of where the mass was resected, or where the radiation port actually killed a lot of the cells. So the thing we have to, the first question we ask whenever we're doing a study is, does it get into the brain? In other words, does the drug get into the brain? And that's the question we need to ask. And you can answer that question fairly simply. There are PAMPA assays, there are a variety of different assays that give you an idea of how something crosses an endothelial barrier, a normal endothelial barrier. And so there's a way to answer that question. We try to avoid the drugs that don't cross the blood-brain barrier because, I mean, we're, pushing a rock uphill here. So essentially, the issue is that we'd like to be able to use the drugs that either get into the brain nicely or are very effective but can be engineered by medicinal chemists to make them more permeable to the blood-brain barrier.
And just as a follow-up, I'm guessing that's the problem with targeting those EGFR amplifications that you see so frequently in the classical GDMs. Really, the anti-EGFR therapies we currently have really don't cross that barrier, right?
That's correct. Things like alatinib and gefitinib, yeah, those drugs don't really get in. They get into where the bulk of the tumor is, but that's probably going to be mostly dead stuff anyway. They don't get to where the cells that actually come back and produce the recurrent tumor are lurking. So that's why I'd say more than 2/3 of the small molecule inhibitors are not able to cross the blood-brain barrier in physiologically relevant levels.
I had a question over here, I think. Yes, please.
I was wondering if development treatment for cancer is different for children versus adults. Are the avatars still applicable? Metastasis is different. And for T-cell therapy, do you see differences because the virus is still intact, so you have more robust T-cell response?
So the question is whether or not there's a difference in pediatric and adult patients with regards to the various types of therapies that were discussed here. And so essentially, we have pediatric avatars as well. I just didn't talk about them. And they are very informative as to what works and what doesn't work. Pediatric tumors are really a horse of a different color, as it were. The primary tumor that kills children is medulloblastoma, and that's actually derived from a neuroblast progenitor and not from a glial tumor. So it really is like the other type of cell that is in the brain. And those require a different approach in terms of therapy. But the principle is the same. Instead of putting the avatar tumor in the cerebrum, the big part of the brain, we put it in the back in the cerebellum where medulloblastoma normally arises. And as your real estate agent probably told you, it's location, location, location. That's the only thing that really matters. And I think in terms of how these avatars are set up, that's what you have to do. If you're working with ovarian, it needs to go into the peritoneum. If you're working with lung, it needs to go into the lung. Depends on the cancer that you're working with. You need to put it where it likes to be.
In terms of the T-cell therapy, for years it was thought there was no immune response until about the 80s when J.P. Allison and those guys were doing their work. And the nice thing was, yeah, there are T-cells there. I think it's more or less that the fact that there's T-cells that are responding to the tumor. The problem is the T-cells, once they get to the tumor, they're inactivated by the tumor. So changing that environment, even in the spleen, the lymph node, tumor site, is the key. And so that's why we have to reactivate those T cells. And it has been recently discovered that you can take these inactivated T cells and convert them back to killers. It's just a matter of waking them up. So I think that's the issue, really, in terms of T cell therapy.
And just to address the metastasis side of things, people are beginning to use the avatars, and Alana Whelm, to the best of my knowledge at Utah, has made a series of breast lines that the avatars metastasize to the same organs as the parent, and that includes bone, which basically, except for one sporadic tumor type in mouse, the animal models have not replicated those. So there's promise there, but it hasn't been explored particularly heavily to my knowledge.
Okay.
This is for Dave. So all the sequences they do now on the expression or sequencing of tumors, that typical sequence you show all mitochondria. So the question is, can we follow the mitochondrial sequence and things? I've not done any of it, and so I'm counting on people who I've talked to. For the most part, anything that's not aligned to the various chromosomes is thrown out. And there are some larger groups at the Broad, TGen, and so on. So some. And it turns out that for what it's worth, the folks at Life Technologies in their Torrent system are actually making mitochondrial sequencing chips because they are beginning to see a role of the mitochondria. So I do think it's going to be an accumulating a bit of data. I'll try and paraphrase for the folks who are online. The idea, I thank you for liking the idea of maybe just controlling things as opposed to eliminating, making glue from the horses instead of anything else. I'm going to slightly disagree with the popularity of that. What I'm seeing when I talk to patients, various survivor groups and so on, is patients are warming up to the idea of just give me a good quality of life. And if you can extend my lifespan to something resembling normal, it's not so bad that the tumor's there. So there's the group that's actually said to make metastases a chronic rather than lethal disease, and they're willing to accept that. I think everybody would appreciate, let's just get the sucker out of me and not have to deal with it, but it's a baby step.
And I would have to add that the pharmaceutical industry would love to have drugs that convert cancer from an acute disease to a chronic disease, because that means that patients are taking the drugs for the rest of their life as opposed to short-term uses. You know, Gilead is charging $84,000 to cure your herpes virus, your hepatitis virus infection in 12 weeks. You know, and so the issue is that, you know, they can't, you know, at $84,000, they are barely making their money back on that particular drug, you know, considering what they paid for the company that actually came up with it. So, you know, the issue is that if you can convert cancer to a chronic disease, then you've got something like diabetes or hypertension, you know, forever, and those are traded forever. And essentially you can live with it, and you've got good quality of life at the same time. So, you know, we may be trying to go for the home run, but it might be that a double is just as good for most patients.
Of course, the evolutionary aspect, every time we usually tinker with the environment, you know, like in terms of mice and plants and snakes, we get rid of something, but something worse develops. That's kind of bakeries, some of the things some of the treatments that people adapt to are not that good.
With the evolution of tumors, evolution requires a division and a passing along of the genetic material to the progeny that somehow a change. So if the idea could be taken for a dormant cell, means it's not generating progeny because the DNA is not being replicated, and that would obviate evolution, at least in theory. I have absolutely no doubt that somehow, some way, as we age, somehow the microenvironment is going to change sufficient that some signal will go to those cells eventually that have been held in a non-dividing state. that will trigger something to evolve. But if we can buy a little bit of time, then Yancey and Greg can.
Data that you discussed today, except for my single paper, which was it is
Well, one of the problems you have to think about in that regard is that a lot of the things that we're using today to treat patients with cancer are carcinogenic. You know, we see a lot of tumors that develop in long-term survivors who had meningioma, and we were treated with radiation or some other disease and treated with radiation. We see those tumors coming back in the brain or arising in the brain de novo. And a lot of the chemotherapy that's being used will produce leukemias and other types of disorders eventually. So, we've got to find a smarter way to do this and not just try to dicker with the drug dose or the radiation dose and combinations of those two. We've got to find things that really target the tumor in some specific way.
If you don't mind, along those ideas, during your talk, when you were talking about the P27, it was occurring to me that because it's having an effect on the metastatic behavior or the migratory behavior and on cell cycle, if there's some way that you could sort of drive the P27's cell cycle inhibitory machinery while blocking what it's using at the membrane with a single or a couple of agents, you might have the double whammy that everybody's looking for. Have you all thought to do that?
You know, the first thing on the back that is the patients. We can mention and that's one of the reasons why we should be removing this activity that will be out of the cytosol.
P27 into the nucleus, then,
I had one question about potentiating the antigenic appearance of tumors. The abscopal effect is when you radiate one tumor, you can get a response on the distant metastases. Have you, Dr. Gregg, have you considered anything about potentiating the effects that you're seeing with radiation?
Actually, we were doing similar things like that when I was doing the T-cell transfers. In some cases, we would irradiate the animals. I did a few studies like that, and then transfer the T-cells in. The results were somewhat similar, so I'm not really sure what's happening there, but yeah, it makes sense. You would irradiate, that would generate lots of antigen, lots of debris, and mature dendritic cells, and you would get a nice T-cell response. And we saw that, but it just didn't seem to add to the effect of the T-cells. Probably because maybe the regulatory populations weren't damaged enough or significantly reduced. I did a couple of studies where I looked at a couple of the markers. I don't know if you can say they're specifically for regulatory populations, but when I did look at those, they weren't severely ablated. So not sure if that's the answer, but yeah, that's a good point. So.
Well, for what it's worth, there were a whole lot of studies done in the late 60s and early 70s by Bonnie Miller, who was actually taking a number of mammary carcinomas derived from the same tumor and putting them on different sites on the sides of the body. And the tumors were communicating with each other and regulating the metastatic ability, the angiogenic ability, and the like for tumors at distant sites. So besides the immune system, there are other things that are being regulated, and even though that work is 40 years old, it's not been explained. So there's the best phrase I've heard for it was from Gloria Hepner, who referred to them as tumor cell societies. Unfortunately, they're all pathologic societies, but they're clearly gangs in the hood as opposed to a civilized, well-behaved, ordered society.
So, before I know this last question, Greg, Dr. Greg,
This is Steve Russell's work with the measles virus, in which they've modified the virus to target it to specific receptors that are overexpressed on certain tumor cells. So he's been doing this for quite some time, and very effectively, I might add. They've treated ovarian cancer with it, and this is the first time they've had a quote cure in which all the tumor cells seem to disappear from the bloodstream. Virus therapy. Yeah, go for it.
Do we have another question? Anybody that got left out? Well, if not, I'll take the liberty of concluding the session. The conference will resume tomorrow morning with the space biology part of the conference. Please come about 7:30. There'll be a continental breakfast outside with some coffee, and the meeting will actually start at 8:15. And the start with 8:30 is the keynote address by Joel Kearns on the value of microgravity research. Thank you all for being here. Thank you for hanging out with us to the bitter end. I think the discussion was very, very good. And again, let's thank our speakers from the afternoon session.